vitamin D, that's calciferol, and you find it in fish, in milk, in egg, in liver. Then it is needed for strong bones, formation of strong bones. It is formed in the skin by sunlight. Vitamin D is also formed in the skin by sunlight. Then it helps in teeth formation and development. And deficiency of vitamin D will lead to rickets, that's the curving of the bones of the leg. Then osteomalacia is also a bone problem. Vitamin E, ergosterol, you find it in green vegetables, in your egg, in butter, and in liver. It leads to promotion of fertility in animals. It helps the productive system. Then deficiency of vitamin E leads to sterility or premature abortion. That is, there will be infertility or premature abortion. Then the next vitamin is vitamin K. That's the last one we'll be talking about, and that is phylloquinone. That's vitamin K. Then you find it in fresh vegetables, e.g. your cabbage and spinach. Then vitamin K aids blood clotting. That is, when there is wound, the blood easily clots. But when it is deficient in the body, you have what is called hemorrhage. That is, inability of the blood to clot. When somebody gets injured, in case of an accident, the blood does not clot on time, the person keeps losing blood. That is a condition called hemorrhage. So we move on to talk about another class of food called water. Water does not give energy to the body, but is essential in all life processes. About 60 to 70% of the body contains water. Then about 90% of the blood plasma is water. So you see that water is a major component of our body. Then it is composed of hydrogen and oxygen. Various sources of water include rivers, streams, well, tap, rain, pond, then from our food. So we have these sources of water. What importance does water have? Since it's not energy giving, it aids in the metabolic activities of the body. Water aids digestion, food digestion. Also, it helps to maintain the body temperature, make sure that there is a balance. Water also transports materials in plants and in animals. It transports materials. Also, water aids cooling. When water evaporates from the body of the animal, there is a cooling effect that it has. Also, it is responsible for evaporation of metabolic waste from the body, e.g. urine. Water is responsible for the evaporation of urine in the body, removal of them. Then water maintains the osmotic content of the body. There is a balance that water brings to the body. Also, water is a solvent for soluble food substances in digestion. Water aids digestion. Water is also helpful in seed germination. No seed germinates without water. Water is a major component of the plant and the animal body. About 75% of a man's body is water. So the last class of food that I'll be talking about is the roughages. Roughages are food materials that consist mostly of fibers, which add to the bulk of materials in the large intestine. Even though roughages do not supply much nutrients to the body, they help to stimulate the body, the muscle of the intestine, so as to push feces along the intestine for easy defecation. They help in easy defecation, that is, they help to push waste out of the intestine. An example of roughages, we have vegetables, we have apples, we have maize, and fruits generally. They are roughages that help in the removal of waste from the body. Lack of roughages in food results in constipation. Lack of roughages in food will result in constipation, that is, the food will not digest properly and the person will be very, very uncomfortable. Symptoms or absence of roughages, I said earlier, constipation, discomfort, headache. Those are symptoms of absence of roughages in the body. Balanced diet. Balanced diet is a food that contains sufficient amount of all the classes of food substances required by man. A balanced diet is a food that contains sufficient amount of all classes of food that is required by man. And balanced diet is important to be eaten at all times because it helps you to stay healthy. One, it prevents diseases and the deficiencies of the food substances. Then balanced diet promotes growth. It also gives the required energy. Balanced diet prevents malnutrition and increases the body's resistance to diseases. Deficiency diseases. What are deficiency diseases? These are diseases that are caused by lack of certain food substances in the diet. And examples of deficiency diseases include kwashiorkor. Kwashiorkor is due to lack of protein in the body. Then scurvy. Scurvy is due to lack of vitamin C. Then pellagra. Pellagra is due to lack of niacin, that's vitamin B3. Night blindness is due to lack of vitamin A. Then goiter is due to lack of iodine or vitamin D. So we want to talk about food tests. Food tests. How can we test for food? That's a lab work now. When we get to the laboratory, how can you know that this particular food is present in this food substance that you are presented with or not? And first, we'll be talking about test for carbohydrates. How do you test for carbohydrates in food? Or how to test for glucose or the reducing sugar. To the 5 ml solution of glucose, add equal amounts of Ferrin solution A and B. We have a solution called Ferrin solution A, then we have Ferrin solution B. So you add equal amounts of Ferrin solution A and B to 5 ml solution of glucose. Then you eat, that is, you add it. Maybe you place your beaker on the Bunsen burner with lights, then you eat it gently. After a while, you discover via a brick red precipitate, and that shows the presence of glucose. A brick red precipitate, when heated, when Ferrin solution A and B is present, shows or indicates the presence of glucose. Another test for carbohydrates is the test called the Benedict test. Add 5 ml of glucose solution and 5 ml of Benedict solution in a test tube. Then you allow it to boil. You boil it. 5 ml of glucose solution, 5 ml of Benedict solution in a test tube. Then you boil it. After, you allow it to cool. Then, in the cooling process, you will discover a red or orange precipitate that is formed in that test tube. That is also showing or indicating the presence of glucose. Now, test for starch. How do you test for starch? That is also called iodine test. You place a few drops of iodine solution to a starch solution. A starch solution, any starchy food. So you add a few drops of iodine solution. The moment you add the few drops of iodine solution, there is a change of color to blue-black color. That shows the presence of starch. Then also, you can add a few drops of iodine solution to the surface of yam or potato. You know, that's starchy food. So when you add iodine solution to the surface of yam or potato, you also find a blue-black color, which indicates the presence of starch. Now, test for sucrose or maltose. That's a non-reducing sugar. You put 5 ml of sucrose or maltose solution into 2 different test tubes. 5 ml of sucrose or maltose into 2 different test tubes. Then you label them A and B. To the test tube A, you add 5 ml of Benedict solution and boil for 5 minutes. When you do that, you discover that there is no precipitate that is formed. Now, to the test tube B, you add a few drops of dilute hydrochloric acid. Then you boil this gently for 2 minutes. the hydrochloric acid will hydrolyze the sucrose or the maltose to glucose sugar. Then after that, you add 5 ml of the failing solution A and B and boil gently for 2 to 3 minutes, and then you allow it to cool. You will discover that there is a brick red precipitate, and that shows the presence of a reducing sugar in the hydrolyzed solution. So it tells you that sucrose or maltose is present. Test for protein. How do you test for protein? We have 3 types of tests. We have the millions test, we have the Burette test, and the xanthoproteic test. The million test, about 5 ml of a protein solution, for instance egg albumin, you add 5 ml of millions reagents and you boil it for 3 minutes. When you have a solution of protein food, for instance the albumin of the egg, you add 5 ml of million reagents, then you boil it for 3 minutes. You discover a brick red precipitate is being formed. So that indicates the presence of protein. Then we'll move on to the Burette test. To 1 ml of the protein solution, you add 1 ml of 20% sodium hydroxide, then a drop of 1%